Bonjour tout le monde, et merci Sandrine pour l'introduction et l'invitation. As you can tell, I'm much more comfortable in English, so I'll continue in English, but feel free if you'd like to pose a question in French. I should be able to understand. So today I'm going to be talking about one of my favorite topics, which is methane, and using isotope measurements of methane to better understand emissions of methane to the atmosphere. And I'll be focusing on permafrost environments at a variety of scales, and some of them are environments that many of you in the Centre du Sud Nordique are familiar with, such as the SAS2 Valley near Wapmagustui, Kuujjuarpik. Before I launch into the research, I just want to do some acknowledgments. And first and foremost, the students in my group, so Regina Gonzalez-Macuel is a PhD student who has contributed to a lot of this work, especially what I'll be discussing in the second 2/3 of the talk, and has been doing really excellent work. And also there's a team of 3 undergraduate students, Emerald, Senga, and Dawson, who contributed quite a bit to what I'll be discussing in the first part of the talk. I also need to thank the funders for this research, so funding from the FRQNT and INSERC, as well as the Trottier Institute for Science and Public Policy, which is a really great institute at McGill. And then through international collaborators, I've had funding from the UK Natural Environment Research Council, and Regina's PhD has been partially funded by the Mexican federal funding agency CONACYT. And then finally, this work began at Caltech, where I was funded very generously by a postdoctoral fellowship. There's a long list of collaborators, some of them members of CEN, who I need to thank. I'm not going to name them all, but a number of people across Canada, the United States, and Europe have contributed to this research. And then finally, very importantly, some of this work was greatly facilitated by use of the CEN facilities, especially at Wapmagustui, Kuujjuarpik. And I want to acknowledge that this work took place in the territory of the communities of Kuujjuarpik and Wapmagustui. And currently, as I'm in Montreal, I should also acknowledge that I'm on land that is the traditional gathering place at the Haudenosaunee and the Anishinaabe. Okay, so today I'm talking about methane, and methane is important for any number of reasons, but especially as a greenhouse gas. I think many of you are familiar with this, but it is about 35 times stronger than carbon dioxide as a greenhouse gas on a per molecule basis. And therefore, it's very important for anthropogenic global warming. We think it accounts for about 25% of anthropogenic global warming. And humans have had a very large impact on methane in the atmosphere. There's been a huge increase in methane concentrations in the atmosphere during what we would call the Anthropocene. However, we don't understand methane as well as we do carbon dioxide, and especially the balance of sources and sinks that are causing methane to increase in the atmosphere, as well as smaller scale variability, such as the leveling off of atmospheric concentrations during the 1990s, and then the subsequent increase after that. In addition, we don't have a great understanding of what might happen with methane emissions in the future and the role that feedbacks to global warming might play in that. And a really important feedback that has gotten a lot of attention is the permafrost carbon climate feedback. So, as I'm sure most of you are very familiar with, there is a very large amount of carbon stored in permafrost soils around the circumarctic, roughly speaking about twice as much carbon as there is in the atmosphere. And importantly, most of this carbon has been isolated from the atmosphere for thousands to tens of thousands of years. And so when it is released and potentially put back into the atmosphere, it represents a net input of carbon into the active global carbon cycle. So we can essentially consider it like fossil carbon. And the feedback loop that I'm referring to is fairly straightforward. As we increase temperature in the Arctic, we thaw permafrost, and then the carbon that is stored in permafrost becomes destabilized, and some of that can be converted to carbon dioxide or methane and released into the atmosphere, leading to further warming. And some parts of this loop are quite well known. The parts that are less well understood is exactly how much permafrost carbon is converted to greenhouse gases, as opposed to being sent to some other long-term carbon reservoir. And then what percentage of that carbon that is released is in the form of carbon dioxide or methane. And these are important questions to ultimately understanding how strong this feedback may be and when it will start really impacting the Earth. And something that we realized over the past decade or so is that lakes and ponds are a really important component of this feedback. So this is details data from a paper published by my colleague Katie Walter-Anthony, but many members of CEN are also working on this question. But in general, we find that lakes and ponds are hotspots of greenhouse gas emissions in permafrost landscapes. So when you look at upland soils, you see fairly limited greenhouse gas emissions dominated by CO2, and in some cases, net carbon uptake from the atmosphere. But when you go to these permafrost thaw lakes, either places where you have expanding lakes or new lakes, you see very large greenhouse gas emissions, and much of that is dominated by methane. So we think these are zones of, these are hotspots of greenhouse gas production and emission. And I'm not showing these data here, but when we look at radiocarbon measurements, we also see that much of this carbon is quite old, indicating that indeed it's coming from thawing permafrost. Still, we have a lot of questions about exactly how, why, and where we get these emissions from lakes and ponds and permafrost environments. And there's a lot of questions that remain unresolved. A lot of these are, again, about how methane is produced and then how it is emitted to the atmosphere. And so I'm going to talk about both methanogenesis pathways or biochemical pathways of methane production. And the two key pathways that we often talk about are the hydrogenotrophic pathway, where carbon dioxide is combined with hydrogen to form methane and water, and the acetotrophic or fermentation pathway, where a relatively large organic molecule, acetate, is broken down into both CO2 and methane. There are other pathways. Generally speaking, we don't think they're very important in a quantitative sense, although there's been maybe some revision of that, and it's an interesting question. And then there's emissions pathways, how the gas that is formed is released into the atmosphere. And there's three key emissions pathways that we often talk about. One through plants, and through specifically plants that have these specialized gas conduits called parenchyma. I'm not going to talk about this. I don't know as much about this. And it's, I think, of the three, the least well studied, but is definitely important. But I'll be focusing more on ebullition, or the release of bubbles that form in sediments that, when they build up enough pressure, exit through the water column and then straight into the atmosphere. And then the diffusion of dissolved gas. So we have dissolved methane in the water column, and through both diffusion and advection, this is eventually released into the atmosphere. And so three key questions that I've been focused on that I'm going to be talking about today are, when we have old carbon released from permafrost, is it rapidly converted to methane by these biochemical pathways, or because by nature of its age and possibly its refractory chemical composition, is it relatively slow to be converted to methane? Are there different biochemical or emissions pathways that are more likely to release permafrost carbon than others? Are some of these emissions or production pathways more important when we think about permafrost? And then finally, from a larger scale, are there signals that we can use to detect increasing high latitude freshwater methane emissions in the atmosphere? If this permafrost feedback is accelerating, can we see signals of this in the atmosphere? So, as Sandrine mentioned, I'm an isotope biogeochemist primarily, and the tools that I employ to try to answer these questions are primarily measuring rare isotopes of methane. So as a very brief primer, an isotope is an atom that contains a differential number of neutrons and therefore has a different mass. And so when we think about methane, there are two elements that we can consider isotopes of: carbon and hydrogen. And by far the most common form of methane contains carbon-12 and hydrogen-1, about 99% of methane. But there are measurable quantities of methane that either contain carbon-13 or deuterium. And measuring the relative abundance of these rare isotopes can tell us a lot about biochemical pathways, about oxidation, and potentially about other processes as well. So this is a somewhat famous figure in the world of methane isotope geochemistry from a Canadian colleague, Michael Whitaker, showing how different types of methane or different sources of methane have different isotopic compositions, and microbial methane that we would see produced in lakes and ponds is always has a relatively light isotopic composition. So it has less carbon-13 and less deuterium than, for instance, methane produced in high temperature environments. And then potentially we can also differentiate, for instance, hydrogen-trophic methane from methane produced by fermentation based on isotopes as well. I'm a little bit more skeptical about how well this works uniformly in practice, but it has been applied quite widely to do this in the past. Our general understanding of methane isotope biogeochemistry hasn't actually changed that much since this was first published 20 years ago. Although we have, it has been evolving, but I think the general kind of scientific consensus remains, this remains a pretty good summary of that. But there has been a really big acceleration of methane isotope measurements recently, in large part because of new technologies such as the Picaro carbon isotope analyzer that use laser-based methods and has really brought down the cost and difficulty, specifically making this delta 13C measurement. And so we see a lot more data on that measurement in particular. But what there's less of is the other isotope measurements. So it's the end of ski season, or maybe ski season is over, unfortunately. But I'm using this kind of notation from Ski Hill to kind of refer to the relative difficulty of the different isotope measurements. And so to measure carbon-13 is relatively easy, especially now with these laser-based methods. Measuring deuterium has been done for a long time, but remains more challenging, so we're going to call this an intermediate measurement. It's somewhat more difficult. Measuring radiocarbon, so the radioactive isotope of carbon, is substantially more challenging, especially just because there's so little radiocarbon. And so we need very specialized measurement methods. We'll call that an advanced measurement. And then finally, there's a very new measurement where we're actually looking at molecules that contain two rare isotopes, so 13C and deuterium, or two deuterium atoms. We call this clumped isotopes because two of our isotopes are clumped together. And I'm calling this experts only because it's really only been developed in the past few years. And we're still both understanding how to make the measurement, but also what it means. So my kind of key premise for this talk, which I'll be covering a lot of different elements of this, is that using this wider kind of set of isotope measurements can be very useful in understanding these processes of permafrost thaw and methane emissions. And I'm going to go over 3 case studies focused on these more challenging isotope measurements. And these are going to go from intermediate to advanced to experts in my Ski Hill terminology, and also going kind of in reverse order of the actual chronology of the research. So from the most recent material that's been published to some slightly older material to something that was started in my postdoc and has been continuing at McGill. And specifically, I'll be looking at methane hydrogen isotopes at a big scale, how this can tell us about high latitude methane emissions into the atmosphere, and then looking at a more local scale, how methane radiocarbon measurements can help us identify differences in methane carbon sources by emission pathway, season, and pond type. And then finally, looking at this clumped isotope measurement, seeing what we think it can tell us in permafrost environments, and what I think it's telling us is about differences in the rate of methane production and why that might be interesting. Okay, just very quickly, this is not a very fun slide, but just the notation of stable isotopes is important to have a quick review of. So when we measure rare isotopes, we always measure them as a ratio to our most common isotopes. When we talk about, for instance, hydrogen isotopes, we're measuring the relative abundance of methane containing deuterium to methane with no deuterium, and likewise for carbon-13 and carbon-12. But then we always reference this to an international standard and then calculate this delta value. So that's what I'll be referring to today. And I'll be using delta 2H and delta D interchangeably. The isotope community is kind of in a shift. We're moving away from delta D towards delta 2H. They mean the same thing. I'll probably say delta D more because it's easier for me to say, but you may see both terms on the slides. So one interesting aspect of methane delta D values that sometimes gets overlooked is that they're strongly influenced by the hydrogen isotope composition of environmental water. And we've known this for quite a while. This is data from what's been a very influential study by Susan Waldron, showing there's a nice linear correlation when you look at freshwater methane between the hydrogen isotope composition of water and the hydrogen isotope composition of methane. And this is potentially very useful because the isotopic composition of precipitation varies very strongly by latitude. And so we can see this on this plot. We have very negative values in the high latitudes and much more positive values in the tropics. And so because of that, we would expect, sorry about that, that our methane delta D would also potentially vary by latitude, and we'd be able to detect a different signal of methane delta D in the high latitudes. So I worked with a team of undergraduates to compile an updated global data set of freshwater methane delta 2H values, and I'm putting global in quotation marks because, as you can see, we have a really biased distribution of data. There's a lot of data from North America, some from Europe, but very, very little data from the tropics. And this is a real important data gap, and I think something that people should focus on more is collecting a lot more data from the tropics, which is actually the largest region of methane emissions. We need to understand this better. However, I think we get enough latitudinal variation that we can characterize this relationship between water and methane delta D values. And the brief summary is that indeed we observe a correlation between these variables that is significant and that is broadly consistent with the previously published relationship, but it is also somewhat different, especially in high latitudes where we now have a lot more data, and we see overall more enriched values in high latitudes that would have been predicted by the previous relationship, which is shown here in blue. So then the next question is this relationship strong enough that we can actually differentiate methane by the latitude in which it's produced? And the short answer is yes. So these box plots are showing the distribution of methane hydrogen isotope values in low latitude, mid latitude, and high latitude environments for all sites, for wetland sites, and inland water sites. And what we can see is that there's clearly a difference, and it's a statistically significant difference when we compare high latitudes with other latitudes. So we can use methane hydrogen isotopes to differentiate methane that originates in the high latitudes from freshwater environments versus other latitudes. People have previously proposed that we can also do this with carbon isotopes for somewhat more complicated reasons that I'm not going to delve into. Our analysis suggests that this is more equivocal. It's less clear that this is actually the case. We see some latitudinal variation, but it's not consistent between different ecosystems. So I think this needs more study. There's a lot more data. We didn't sample all the measurements by any means, and I think someone's going to have, maybe me, maybe someone else, will have to dig into this and really compile a lot of data to fully answer this question. The next question that I wanted to tackle was whether this latitudinal difference is actually enough to create a detectable signal in the atmosphere. And so to do this, I worked with a team from the University of Bristol, have a lot more expertise in atmospheric methane modeling. And the first step was to create a map of what we expect methane delta D values to be based on precipitation delta D values. So that's what's shown on this panel here. And then we use that to make a prediction of what the methane delta D value in the atmosphere would be in the southern hemisphere, shown here in red, globally, and in the northern hemisphere in blue. And we see two lines here. The dashed line is the baseline emissions model based on our current best estimate of where emissions are coming from and the current distribution of freshwater emissions to the atmosphere. And then the solid line is if we shift 8% of global emissions from the tropics to the high latitudes. And that's within the realm of current uncertainty. And it's also, what we might expect if we have more emissions from the high latitudes as we have ongoing permafrost thaw. And the short, summary of this is that there is a detectable shift. It's about 5 per mil in the northern hemisphere, which is definitely within analytical uncertainty. And so we should be able to detect, with all other things being equal, and that's an important caveat, a shift towards more high latitude emissions based on methane delta D measurements. Now, currently there's not a systematic global set of methane delta D measurements to trace this kind of process in the atmosphere. This is in contrast to delta 13C, where there's been much more of a routine and systematic effort to make that measurement. And the key difference is really that, the methane delta D measurement is more challenging, and there has also been questions about its overall utility. But I think these data, along with many others, are showing that it is in fact a very useful measurement. It is important to do more routine measurements. And so hopefully new technological developments such as these laser-based methods for making this measurement that have recently come out will allow us to make this more frequently and have more data to work with. Now, as you probably would noticed and maybe you wanted to comment on, there's a lot of residual variability when we look at this relationship between water and methane delta D. And there's clearly other processes and other variables that are very important for this. What's less clear is exactly what processes are most important for controlling this in freshwater environments. But I think this residual variability, when we look at a given site, can be really interesting and very powerful for understanding the processes controlling methane generation. And as an example of this, I have data from a collaboration I worked on with Isabelle Lorient and her PhD student Vilmontes, looking at ponds from Violet Island. And we see really huge variation in methane delta D in these ponds, with especially negative values in these ice wedge trough ponds, which are shown in the red and orange triangles. And we still don't really understand why we get such negative values in these ponds, but it's really quite an interesting signal. Personally, I don't think it's related to either differences in biochemical pathway or methane oxidation, and something else. Something I'm quite interested in is whether it could be a function of metabolic reaction rates. But we don't know enough about this to put a finger on it, but I think it's a really interesting axis of variability that we can explore more to try to understand why methane generation is different in these different freshwater environments. Okay, so I'm reached the end of talking about hydrogen isotopes, and as I mentioned to Sandrine yesterday, I'd be happy to take questions kind of in between if there are any, because I'm now going to talk about a different isotope system, or I can just keep going. So if anyone has a quick question, feel free to let me know.
So people can either raise their hands electronically or write in the Q&A. And up to now, I don't see anything of that.
I'll just keep going. I have a lot to cover, but feel free at any point really to stop if you do have a question you'd like to ask. Okay, so now I'm going to move on to talking about methane radiocarbon measurements. And so I'm sure most of you are familiar with radiocarbon. It is a cosmogenic radioactive isotope produced in the atmosphere by cosmic rays, and it's fixed in the biosphere through photosynthesis, and then it decays with time with a relatively short half-life. And so what it really is very useful for is a clock telling us how long it's been since, you know, a carbon reservoir was fixed from the atmosphere. What's also really useful about it is when we measure it, so we measure the ratio in our sample relative to a sample from the year 1950, and then we correct that for the delta 13C value of our sample. And because of that, we're correcting for any biochemical fractionation. So essentially, the radiocarbon value is transferred conservatively from one carbon reservoir to another, and so it's a really good tool for identifying carbon sources, for instance, of methane. And then we can convert our fraction or fraction modern value into an estimate of time using this term 14C years. It's good to note that this is not actual years, and so we have to take into account there's uncertainty there. But because carbon in permafrost has been sequestered from the atmosphere for a long time, it's very easy to distinguish, for instance, methane that has been produced from recently fixed actively cycling carbon, which should have a fraction modern value greater than 1, and then methane from permafrost carbon, which will have a lower fraction modern value, usually less than 0.8. And 14C measurements are also getting easier. And so this is, you know, they're notoriously expensive and you need very large samples, but this is gradually becoming somewhat easier, in part because of new measurement technologies. So for instance, there's a new instrument called the MICADIS, or Mini Radiocarbon Dating System, and one of these has recently been brought to the University of Ottawa, the Lalonde AMS lab. So this is hopefully going to reduce the costs and sample sizes needed and hopefully kind of accelerate radiocarbon research in Canada. And then also through sample preparation methods, we're able to now better separate gases like carbon dioxide and methane for 14C analysis. This is a picture of Regina sampling a very large quantity of water at one of the ponds in the SAS2 Valley so that she can make a dissolved 14C measurement. And this is like essentially a very large head space equilibration method, but you know, a few years ago this would not really have been possible, but now with improved measurement and sample preparation techniques we can make this measurement. And so Regina's been focused on making a very extensive set of 14C measurements in thermokarst ponds in the vicinity of Wapmagustui, Kujawarapik, trying to look at, broadly speaking, what is the variation in carbon sources to methane and other carbon fractions across these ponds. And so she's looked both at the Lethalsa lakes or ponds in the Quack Valley. So these are formed in mineral soils, as well as the Palsa Ponds in the SAS2 study area. And I think many of you are familiar with these study areas. And in addition to looking at the differences between these two types of ponds, she's also been looking at the two emissions pathways, so ebullition versus dissolved methane. In these environments, dissolved methane is by far the larger flux, but we want to know whether these two different fluxes are being fueled by different carbon sources. And then she's also looked at differences between winter and summer. Are there seasonal differences in carbon sources? And then finally comparing methane with other carbon fractions, including dissolved organic carbon, which I'll be talking about, to get a better understanding of what might be the key sources of methane production in these environments. So I won't be able to cover all of this work. It was quite extensive, as I mentioned, so I'm going to focus on a few key questions. The first of these is ebullition versus dissolved methane. And so what Regina found, looking at this plot, is that dissolved methane in the Palsa lakes is, relatively speaking, young. It's not modern carbon, but it has an average 14C age of about 250 years, whereas the ebullition methane, the methane coming out in the bubbles, is much older. It's about 1,500 years. We clearly have a different carbon source with much older carbon coming out through the ebullition methane. And interestingly, in this environment, this is actually older than the age of permafrost formation. So we're seeing the release of carbon that predates permafrost formation in this environment, probably from deep peat carbon. The general result that the dissolved methane is younger than the ebullition methane is consistent with results from other locations in permafrost environments such as Alaska, Siberia, Western Canada. I think also at Violet, although the differences are a bit smaller there. But interestingly, we don't see a difference in the Lethalsa ponds. There, the age is essentially identical, and it's all modern carbon. So there's some difference with these Lethalsa ponds where we're not seeing this input of old carbon into the methane pool. I think probably this may just represent a different sedimentary environment with less deep carbon within sediments that's accessible for methane production. It also may represent that gas transport from deep sediments is more challenging because there's a thick clay layer in these environments. We're not exactly sure of the mechanism producing or preventing the release of old carbon, but it's a very clear difference between these pond types. Another interesting finding was a big difference in the age of dissolved organic carbon and both dissolved methane and also dissolved inorganic carbon. And so we see a very clear signal that the dissolved organic carbon, or DOC, is significantly older than these other two carbon fractions. In this plot, this is shown where we have dissolved methane with the green circles, DIC in the blue triangles relative to the fraction modern of dissolved organic carbon. And all of our data are in this upper quadrant, meaning that these DIC and methane are consistently younger than DOC. And what I think this is telling us is that while DOC is probably still the likely source of carbon for both of these respired carbon pools, methanogens are preferentially using a relatively young and labile fraction of dissolved organic carbon and leaving the rest behind, essentially. And so they're only using the youngest material. And another interesting aspect is that when we look in winter, the DOC values, and to a lesser extent DIC, shift towards older carbon. So we're getting some input of old carbon into the ponds in winter, but this isn't changing the radiocarbon age of the methane. The methane remains constant. And so either, even more so in winter, the methanogens are preferentially using young carbon, or an alternative hypothesis is there's just not any new methane production in winter, and so we're not seeing any shift. It's basically just remaining constant through the winter season. So Regina's been following up on this to get a better understanding of what controls the radiocarbon age of methane in these environments by doing a set of incubation experiments. And this is focused on sediments from two ponds in the SAS2 Valley, SAS2C and SAS2B, where she's taken sediments and incubated them at 3 different temperatures, 4 degrees, 10 degrees, and 25 degrees. And the key question is, or our hypothesis is, that we will have older carbon in the methane at higher temperatures. And the rationale for this is that this older carbon is more recalcitrant, it has a higher activation energy, and so it will become increasingly accessible to methanogens at higher temperatures. So this is still very preliminary data. We're still doing more experiments and trying to figure this out. But there have been some interesting results so far. And so we do see a difference in radiocarbon age as a function of temperature. So the temperature of the incubation is controlling what carbon is being converted to methane. However, it's complicated because in the two ponds, we see a different response. At SAS-2b, which is further from permafrost, we see what we'd expect, where with increasing temperature, the carbon becomes older. But at SAS-2c, we see the opposite. With increasing temperature, the carbon actually becomes younger. So there's some difference in the organic carbon reservoir in these two ponds that is changing the response to temperature. We're still not exactly sure what this is. It's a very intriguing result. But overall, the picture is that methane is produced from younger carbon at faster rates, and I'm not including these data, but it's much faster at SAS-2b than at SAS-2c in the pond that is further away from permafrost and where there's more young, fresh vegetation. So we think this fresh vegetation is fueling increased methane emissions with young carbon at pond SAS-2b. What's further intriguing is that that's different from what we observe in our field samples, where the radiocarbon age of methane, both in bubbles and in dissolved methane, is quite similar between the two ponds. So we're getting something different in our incubation experiments than what we observed in the field, and still trying to figure out why that would be. So I'm happy, after the talk to hear anyone's ideas on that. Okay, so for the final part of the talk, and I'll pause again for a second if there's any questions about the radiocarbon part of the talk. Okay, so the final part is the most complicated, and I probably left myself with too little time to give it justice, but is this clumped isotope measurement. So again, these are molecules of methane that contain multiple rare isotopes, so either 13C and deuterium or 2 deuterium atoms. These are very, very rare, not as rare as radiocarbon, but incredibly rare nonetheless. And until recently, we didn't really have any hope of measuring these separately from our other rare isotopes. But with the advent of new mass spectrometers that are very high resolution, we can effectively separate these analytes from other molecules that have a mass of 18, for instance, water, which is incredibly abundant, that would interfere with this. And so this is done using an instrument like this. There's only about 8 of these instruments in the world. Unfortunately, none yet in Canada. Hopefully that will change before too long. But I did work on this at Caltech, where there's one of these instruments, and I'm continuing to be involved in this research through collaborations. This, we have to add some more complicated notation here when we talk about this measurement. So like with our more traditional stable isotope measurements, we are measuring the ratio of this mass 18 methane, or clumped isotope methane, to the most common form of methane, mass 16 methane. In the data I'll be showing, we're combining these two mass 18 isotope logs. More recently, people have been separating them and measuring them separately. But on its own, this measurement doesn't tell us that much because it's highly dependent on the total amount of deuterium and 13C in our sample. And so what we have to do then is compare it to what we would expect to see if we randomly distributed our isotopes among all of our molecules. We call this our stochastic ratio. It's calculated using this formula. And then we look at the relative abundance of these mass 18 molecules to what we'd expect from a stochastic distribution. And this gives us our measurement that's meaningful, which we call capital delta 18. And what's, the original interest in this was that this is a function of temperature when methane forms in isotopic equilibrium. We expect to have more of these mass 18 molecules at colder temperatures and less at hotter temperatures. And this actually works quite well in a number of settings. So this is a compilation I published a few years ago. It's definitely been expanded since then. But in volcanic settings, for instance, we get very high temperatures from this, about 300 degrees Celsius, about what we'd expect. In natural gas and oil reservoirs, we see temperatures between about 100 to 250 degrees Celsius, also consistent generally with what we'd expect. And in some microbial methane samples, we also get temperatures that make sense between about 0 to 100 degrees Celsius. But when we look at methane in freshwater environments, we get inferred temperatures that make 0 sense, anywhere from 100 to effectively infinity degrees. And so clearly there's something else going on here. And what's going on is that we have a strong kinetic isotope effect. The microbes are actively selecting against these heavy isotopes and shifting this ratio away from what we'd expect given the temperature. And so what we think instead that's controlling variation in these environments is the rate of methane production, which is influencing this, how strongly this kinetic isotope effect is being expressed. And there's now experimental data to back up this idea that the variation that we observe in these clumped isotope measurements is partially, at least, a function of the rate of methane production. So this is a set of methanogen culture experiments using different carbon substrates, and we see a negative relationship between our clumped isotope measurements and methane production rate. And then that fits pretty well with a model based on theory of what we would expect. Now, the model assumptions require further testing, so this could change, and I'm actually actively reviewing a paper that proposes a somewhat different model. But this seems to make sense, and so I think we can use this at least as an indicator of relative differences of methane production rate in natural environments. And what's encouraging is that if we look at the range of methane production rates in thermokarst lake samples from incubation experiments, and also the range of clumped isotope values from northern lake gas samples, we get a range that's consistent with this model. So that's encouraging, at least, that we're going in the right direction. So I've applied this measurement in a number of permafrost environments, both from Sweden in a place called Stordalen Meyer that's been quite well studied. These are not thermokarst lakes, they're glacial lakes, but they're located in a discontinuous permafrost peatland, so they're influenced by permafrost processes. And then a set of lakes that are thermokarst lakes in Alaska, both from the north slope of Alaska in continuous permafrost and from central Alaska in discontinuous permafrost. And when we look at the clumped isotope data from these systems, the first observation is that we see a wide range of values, indicating a wide range, we think, of different methane production rates. And then a second observation, which includes radiocarbon data, so we're going back to our radiocarbon measurements, is that we see a clear linear relationship between the radiocarbon in these same samples and the clumped isotopes. And this is a negative relationship between the fraction modern carbon and the clumped isotope value. And what that is suggesting is that when we have older carbon, we have slower production rates. This makes intuitive sense, I think. Older carbon we expect to be more recalcitrant, and so methanogens are able to produce methane more slowly when they're provided with older carbon. What's interesting is that if we compare the lakes from Sweden, the glacial lakes, with the thermokarst lakes from Alaska, we see overall lower clumped isotope values, which indicate faster rates of methane production in the Alaskan lakes for a given age of carbon, suggesting that there is a difference in this relationship based on environment. And again, I think this makes sense. In the glacial lakes, the carbon that's present in sediments, much of it was not incorporated in permafrost, so it wasn't frozen for much of its history, and so it's probably more degraded and therefore more recalcitrant relative to its age than the carbon from Alaska. And I see there's a question. Let me see if I can access that. Okay, so this is maybe I'll come back to the question because it's about the previous part, and I'll finish up, and then I'll come back. So let me. Okay, so where things get a bit stranger is that when we compare our methane clumped isotope measurements against the observed flux, or in some cases an estimated flux based on bubble trap data, we see results that aren't very intuitive. So looking in Alaska, we actually see a positive relationship between the flux and the clumped isotope values. And this would actually tell us that when we have our highest observed fluxes, we have the slowest methane production. And so that's obviously not very intuitive. And maybe this is telling us that we're not interpreting this measurement correctly. But assuming that we are, I think one way we can interpret this is that in these systems where we have methane produced deep in these talux or bulbs of thawed permafrost, it's relatively slow. We're using old carbon. But then the bubbles are transported along preferential flow pathways. So they don't just come straight up. They have to move along preferential flow pathways. And then eventually they all congregate and come out in one place. And my colleague Katie Walter-Anthony calls these bubble superhighways. And so even if your per cell production rate is slow, your observed flux coming out of the top of the water column is quite high. Whereas in contrast, if you have younger sediments, you may get your flux coming straight out. You're integrating a relatively small volume of sediment. And this is going to give you a low observed flux, even if the production rate is higher. In the Swedish lakes, we just have no relationship. There's no clear pattern. And so again, I think in addition to production rate, other properties that are controlling gas transport are important in controlling the observed flux, and it's the combination of these two things that is ultimately controlling your flux, and therefore we're not getting a clear relationship with the inferred relative production rate. So that's all I have for today. I'll just have a few conclusions, then I'm happy to take questions. And so my big picture conclusion is that using the full range of methane isotope measurements can help us understand permafrost methane emissions at different scales. Paying more attention to methane hydrogen isotopes could help us to discriminate the geographic source of freshwater methane to the atmosphere, and especially increasing high latitude methane emissions. Methane radiocarbon measurements can reveal important differences in carbon sources and ages between different emission pathways and between different permafrost thaw pond environments. And then finally, this new measurement of clumped isotopes, we're still learning exactly what it's telling us, but it has the potential to give us information about different production rates and how that varies across different permafrost environments. Okay, thank you very much. Sorry I went a bit long, and I'm happy to answer questions.